Thanks for inviting me, Charles. I'm going to give a talk here that's going to be in two pieces. One is going to be a PowerPoint presentation, and the second half of it is going to be an online live demonstration of an interactive tool that we've been building for this interaction with big data. So, sorry, how's that? Better? Okay. So the first half of this is a collaboration between myself and Hamid Boulari, who actually was at the ISB with Lee and then came over to the Hutch to work on this project, essentially, that I'm going to tell you about. And so the idea is something like this. If you just imagine say 1,000 patients where you have molecular data, very dense molecular data, and relatively good clinical data, that's a very big data set. But what you'd like to be able to do is somehow visualize that and interact with it in such a way that you can not only learn about the disease, but actually use that data set operationally. And that sounds like a tall order, right? How do you see 1,000 patients with billions of data points per thing? And so the kind of data that we've been working on, you got to start with the data set to start building tools allowing you to do that. And so the data set that's the most obvious one to work on is the TCGA. And although that's what you like in the future, it is something else, but you have to start with something. And so what the TCGA has, just to remind everybody, is it has methylation data across the genome. It has DNA sequencing information as far as copy number across the genome. It has exome sequencing across all the exomes. It also has RNA-seq for expression analysis. So you have that for every patient, and you have some, in the TCGA case, not great, but reasonable clinical data. And so what we wanted to do was come up with some way of taking all that and collapsing it all down into like 2 dimensions, so you can see like a map or the terrain of the disease. And the way we came about to do this, or the way that Hamid initially thought to do this, was to use multidimensional scaling, which, if you imagine 1,000 patients, each pair of patients is compared for similarity. And so, patient A has got the entire genome sequencing or whatever it is, huge amounts of data in one direction. Patient B in the on the 90-degree angle, and then on the 45-degree angle, if they're identity, then you have, of course, it's all identical. And if it's completely different, then there's nothing common on the 45. And that allows you to collapse down each pair into it gives you a similarity score, essentially. And then if you take all 1,000 patients and you, for every possible pair, generate a similarity score, then you can basically collapse it all down into 2 dimensions. Now, of course, you lose a huge amounts of granular data, but what you are able to look at is sort of landscape, like, how similar are these two things? And depending on what kind of data you put in, that's, you're going to get a different distribution of tumors. Of the same tumors. And so after playing around this for a while, the distribution that we liked the best, or the data set that we liked to pump into the system best, gave us a distribution that looked like this. And so what do we got for pointers here? Here we go. And each dot here is a tumor. And if you have 2 dots that are very close to each other, they have very similar, in this case, whole exome sequence and whole copy number across the entire genome. So vast amounts of data. And then, of course, this dot and that dot are 2 different tumors with very different structure, essentially. And what you can immediately see is that this is not a diffuse cloud. You might have guessed, if you were to do this depth of analysis, that you would actually get stuff just diffusely everywhere. But that's not what you see. What you see is 3 pretty distinct clusters, a little bit of a diffuseness here, but there's not a whole lot of stuff in between these, which would argue that although Mitch is absolutely right, there's a lot of heterogeneity in these tumors, the fundamental fact is that there is some underlying recurrent similarity in DNA structure in this population. And this population is a mixture of glioblastoma and lower grade gliomas. So it's grade 2s, grade 3s, and grade 4s, all mixed together. Okay. So the other thing that you can do, many things you can do, but you can color these dots in based on specific characteristics. So, for example, I can color in the lower grade tumors versus the grade 4s with two different colors, and we can see what that distribution really is, what these dots are. And what you see is that most, although not all, of the glioblastomas live in this region. Most of the lower grade tumors live in these two regions. There are a few GBMs in this cloud, but not that cloud, and there's some lower grade tumors that are mixed in with the GBMs. Okay? Now, just going to take a little detour here and say, using a different data set to distribute the same tumors, in other words, we're going to use methylation data now. You know that Peter Laird identified about 1,500 methylation probes that distinguished the CIMP from the non-CIMP tumors. And remember, the CIMP tumors most of them are low grade. They're the ones with the IDH mutations. It turns out that this is a big distinguishing characteristic in the glioma population. And so if you use that 1,500 probes to distribute these same tumors, you get this distribution. And what you find, without going through all the nuts and bolts of it, is that the tumors that are CIMP, or called CIMP, live right here in this relatively tight cluster. The tumors that are not CIMP live over here. And again, if you color in the glioblastomas versus the lower grade tumors, GBM now in red, most of the GBM live in this space. There's a few that don't. And those that don't were already identified as CIMP GBM. And there are some lower grade tumors that are non-CIMP. Okay? So just keep that in mind. Now, we're going to go back to the original distribution of glioma and color in the lower grade tumors by CIMP versus not. In other words, this group of blue versus that group of blue. And when you do that, you realize that these two clouds are CIMP. And this cloud is basically not CIMP. The open circles are the GBM that are not CIMP, and the blue dots are the lower grade tumors that are CIMP. And so why is this important? This is important because CIMPness is absolutely correlated with DNA structure. And CIMP is a methylation pattern. The DNA structure, which is exome sequence and copy number, is absolutely these two, and you can almost draw a line right here, and almost everything at CIMP is in one side. Okay. Well, what did the pathologists think about these tumors before there was any molecular analysis, right? They look under the microscope, they call it an X, right? How good are they at guessing what cloud a given tumor should be in? Well, it depends on what cloud you're talking about. So this cloud right here is frequently called, a tumor in that cloud is frequently called by the pathologist an oligodendroglioma. Not always, but vast majority of the time. The tumors in this cloud are referred to usually as astrocytomas or oligoastros. There's a few exceptions, but not many. And the lower grade tumors that live with the GBMs are usually astros or oligoastros. And so, to be a kind of a cynic, you might say that the pathologists are using a microscope as a biomarker to try to guess what the actual DNA structure of the tumor is. And you could say that the diagnosis of the tumor, if you're really molecular, is in fact this. Are you living here? Are you living here? Are you living here? That's your disease. Okay. Now, we all know in the glioma world that there are many different mutations that are commonly used clinically to try to tell you what things are. And so I can color in the same plot based on various clinical things. So 1p19q, right? Deletion. Where do these tumors that have that deletion live on this plot? So very, very tight. Essentially everything here is 1p19q deleted and nothing else is. What about mutations in ATRX? These are the mutations usually found in lower IDH mutant tumors, lower grade tumors that portend a worse outcome, right? So they are in red, essentially everything that's up here. But it's interesting, it's actually this upper part of this cloud, the lower part of the cloud has very few of these mutations. What about p53? p53 mutations are found here. Essentially everything in this cloud, almost nothing in this cloud. And in this cloud, it's this region of the glioblastoma cloud, or whatever you want to call it, that's p53 mutant, and nothing down here. Now, again, you might say, well, the cloud or the distribution is all coming from mutations like this. That's true, but it's coming from the entire genome, right? So it's not like one gene is going to be causing this. This is really a way of coloring in the distribution which is caused by the entire genome. Okay, so there's a couple other points you made. One, someone discussed this issue of progression, and so from grade 2 to grade 3, is that structured in the genome or not? And basically, the answer to that is no. So, on this side, the SIMP low-grade tumors, grades 2s and 3 in red and green, are completely mixed together. And in the non-SIMP regions, the grade 2s and 3s, which are blue and black, are also not desegregated. And so what this says is that the progression from 2 to 3, and it turns out to 4. Also, the 4s are the SIMP 4s are up in here. They're not in any way segregated, and so the general DNA structure is something that these tumors seem to have acquired early, and if they progress, they don't lose that during the progression. Okay, what about survival? So if you ask the question, these are grade 2s and 3s, and these are grade 2s and 3s, you probably already know this, but it's pretty easy to pull out the survival of these things, and it's very striking that these grade 2s and 3s have outcomes that are almost as bad as a GBM, and these don't. And so the pathologist might call it an X or a Y, but the fact of the matter is if you map it out, you'll actually have a pretty good idea of the behavior of these tumors. And the final one of these, I think, the final, is that this issue of whether the proneural, the mesenchymal, GBMs, classical, and so on, people make a big deal about it. Not that they shouldn't, but you might ask the question, well, is there, some region of these plots that are proneural, mesenchymal, and so on? And the answer is no. And so all these things, if you color in all 4 of the GBM subtypes, they're completely mixed. And so the subtypeness of a glioma is not hardwired into the genome. All right, this is all. Now to be clear, those subtypes are correlated with specific genetic alterations like EGFR amplification or PDGFR and so on and so forth. But those specific things that they're correlated with are swamped out by the overall structure of the genome that these tumors live in. Okay, so there's other stuff. For example, we can use sequencing to display. And I'm not going to go on and on about all this, but I will just show you a couple of snapshots about the kinds of things that we can do and are doing. So if you use RNA-seq, you can take a particular subset of genes, look at their expression profile, and use that as the input data to display the same tumors. And we will frequently do this. This is a panel of the same, every panel here has the same tumors that I've been showing you, but their distribution is created by different data going in to display. And this corner one right here is the one that I've been showing you. And this was the methylation plot that I showed you. But these other things are other things. So for example, here is a subset of genes associated with stemness. And we're looking at the expression level of several hundred of those genes, or immunologic genes, or metabolism genes. And then here, the coloring system is the diagnosis that a pathologist is given. So again, in green is the oligonucleomas, and here are the astros and oligoastros, and so on. So you can actually see that some of these subtypes have patterns that have pulled out, for example, the green ones in one area or the other. And the argument here is that these tumor types do have, at the expression level, something that makes them, that distinguishes them. We can also pick a particular gene and look at its expression quantitatively as the coloring scheme. So you can go to the next level up. And so again, each one of these is, again, the same tumors distributed by all sorts of different, collective data that was piped in to create the distribution. But the coloring scheme is now not it's not quantal, in other words, yes or no. It's actually a gradation of how much. So in this case, T-cell receptor expression, which is a poor man's judgment of how many T cells are actually in these various tumors. Some areas, in some cases, have very few T cells, and others have a lot. Or Ki67 expression, which is sort of a measure of proliferation. And so just as, a lot of this is sort of distributed out here, but here's an example of what's sort of interesting. It's a group of genes associated with stemness, several hundred, that were used to, their expression used to distribute the tumors. And then the dots were colored in by KSD7 expression. There's actually a gradient of proliferation from one corner to the other, where these tumors have a huge amount of proliferation. These ones have very little, and the distribution, again, is stemness, and that sort of makes sense. And then finally, it's a tool. And we've been building this as a tool to look at data, and of course, this is a glioblastoma meeting, but there's no reason for this to be glioblastoma only. This is an example of that same kind of 16-panel figure for breast cancer, where we've looked where the coloring scheme is, luminal A, luminal B, basal, and so on and so forth. And you can see that many of these data sets that we use to distribute the tumors, breast cancer tumors, are clearly getting coloring in different regions, which means they totally correlate with, or there's a correlation between that, that subtype and a particular expression pattern. Okay, so how would we use this kind of analysis? So just imagine this one's in theory, but not really in theory. You have this as a reference database. So you got 1,000 or ideally 10,000 patients where you've got clinical data on them. You've got molecular data that is used to distribute, and we have the ability to take the next patient, your patient in clinic, with that same data set, we can overlay that patient onto the map. And so if your patient turns out to be here, you can draw a circle around that patient, and the tumors that are very similar in the whole data set to your patient are going to be identified very easily. And if those patients are connected to clinical data, you know how those patients actually did. It's not like, oh, it's an oligodendroglioma. It's actually that neighborhood for that specific, regardless of what a pathologist would say about this. The other way of thinking about it is you could, all these clinical trials that people have done and have, 20% response or 10% response, the drugs put on the shelf because they didn't work. Okay, fine. But you could take the patients in that trial and do the molecular analysis and overlay the entire trial onto a reference data set like this. And here I've done that theoretically, where the black dots and the green dots are the patients, but the green dots are the very few that actually responded. And if it turns out that the ones that responded are all regionalized, then you could imagine rerunning a trial only of patients in that area, because now you sort of know how you're more likely to get a concentration of people that are going to respond. So that's in theory. Okay, and that sort of extends upon what Lee was talking about before. All right, so the second thing I'm going to say here in the last 10 minutes, I guess, or something, is to show you the online version of this, essentially. So this is, here we go. See what we got here. This is something we refer to as Oncoscape. And Oncoscape was built at the Hutch by Hamid and by Paul Shannon, who was also at the ISB, came over to the Hutch, went back to the ISB. So this is this kind of a thing. It's online. It's open source. It's actually at GitHub for any of you who sort of are aficionados of that kind of thing. It's being crowdsourced by lots of computational people to add to it. You have to use Chrome, Google Chrome, to use it right now, or at some point it'll work in Firefox, but it doesn't right now. And this is if you type in this in Chrome, you'll get right into it. It is designed differently than some of the other tools like cBioPortal, in that it is a series of WebSocket applications, each one of which is an individual computational program. So you can analyze the data in one, take a subset of the patients or the tumors or whatever, and move them to a different WebSocket and reanalyze it in a different sort of way. I'm going to show you only a couple of these tabs. These are the tabs that we have currently. Other people are working on other tabs. The simplest one, so one thing you have to do is you have got to pick the TCGA data set. We have TCGA right now, all these different tumor types. And so the one I'm going to show you again is the same one that I showed you earlier, which is TCGA brain, which is low grade and high grade together. One of the tabs is this, which is nothing really, but it's an Excel spreadsheet of what's in the data, and it's interactive, so you can you can limit yourself to a particular age or survival or so on and so forth. Another one is this one. So this one is all of the tumors, all the patients in the data set. If you imagine them as horizontal lines, they were born on a particular blue dot there, and these are the years in which that happened. And then time went on, and they eventually got sick, and then they got treated, and eventually they passed away. Okay, but that you look at that and say, well, that's not so helpful. But you can organize this differently, and you can align them all to the day they were diagnosed. So this is all day 0, and in log scale in days, one direction in retro, and this direction moving forward. And you can begin to see that, more or less 2 weeks after surgery, people started getting radiation, they got some chemotherapy, and then things happened, but that's still not that easy to look at. But you can order it by survival, and now all of a sudden you essentially get a Kaplan-Meier plot. And it's interactive, so I can grab hold of a particular subset of tumors that I'm interested in studying, or patients. You can, from a distance, realize how bad the clinical data is really in the TCGA, because there aren't that many people who were treated after they're dead, or before they're diagnosed, I guess, I hope, right? But that's you can just sort of see, this is 1,000 patients, you can just sort of see the errors. But it's interactive, and I'll come back to that in a second. The other tab that I'm going to show you is this one. And so this tab is made up of 2 sides. This side is the same distribution of those tumors that I was showing you in the PowerPoint presentation. So cloud 1, cloud 2, cloud 3. The size of the dot tells you how many mutations are found in that tumor. Small ones, very few, big ones, a lot. And these are the chromosomes. And the dots on the chromosomes are the genes that are recurrently mutated in this subset of tumors. The size of the dot says there's a whole lot of tumors that have that mutation, and so on. But this is interactive, and so I can pick a particular tumor. I can ask the question, I want to know what mutations are in that one tumor. This is a simple one. And what that does is that it produces a series of lines or edges. In red are the genes that are amplified in that tumor. In green are the genes that are deleted, and in black are genes that are point mutated. Okay, so that's fine. That's good to know. But you can also flip it the other way, and you could say, here's one. Turns out it's IDH, so it's a big dot because there's a whole lot of tumors that have that mutation. I can say, well, show me where all the mutations are that have IDH in that distribution. And they look like that. There's a whole lot of them there. You can select all those tumors. I've turned them yellow. I can send that subset of tumors to a Kaplan-Meier plot and say, well, how do those ones I just highlighted do relative to everybody else from a survival standpoint? And obviously they do much better because they're IDH mutant tumors, right? But that's sort of instantaneous, which is sort of neat. You could also look at collections of tumors. So this is a collection of tumors in this region, which are the oligos, and you can say, well, give me the general lay of the land of what kind of mutations do they have, and you can see there's a bunch of stuff happening here, but this turns out relatively not that much. There's several of them that have gains on chromosome 7, as it turns out. A bunch of them, of course, are IDH mutant, but there's not a whole lot else going on in that region of what we would call the oligo cloud, which is to be distinguished from this region of the astro cloud, if you want to call it that, which looks completely different. All right? And in fact, it's so complicated that you have to move things around just to sort of see where they are. But the point is, where things are red, they're always red. Where they're green, they're always green. So these are chromosomal regions that are recurrently gained or lost. And this is a very different type of tumor than the oligo cloud, even though they're both quote-unquote low-grade tumors, they're both IDH mutant, and so on and so forth. But if we go back to the idea that I showed you earlier, which is your patient comes into clinic, and you say, I want to know how this patient's likely to do. You could say this, your patient shows up here in this little cloud here. You say, all right, I want to know how those patients with similar tumors to mine did. You can obviously send that to survival, and it turns out, you know what? They do pretty well, because this is the oligo cloud. In fact, in this data set, they haven't died yet, which is completely different than if your tumor shows up in this area. If your patient's tumor gets overlaid in this area here, and that is really bad, right? So that's a bad neighborhood to be in, regardless of what the pathologist calls it and so on and so forth. But the truth is there are some patients that do better than others, and your patient, you'd really like to sort of say, I'd like to treat it the way that these guys got treated, fully understanding it's a bad outcome, but how did that happen? So with this kind of a data set, instead of sending to that, I can send it to a different tab, which is back to this thing called timelines. So in fact, this is the same tumors that I just pulled off and showed you it was a bad outcome, but now it's interactive, and I can, you can mouse over these and say, well, the ones that did well were treated with blah, 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 and the ones that did poorly were treated with something else, if that's true, if there really is a difference between them. So this is, again, totally interactive. Just a couple last things. There's a PCA thing where you can, pick a subset of genes and ask the question, does their gene expression, distribute the tumors? And sure enough they do. And these are the tumors, and distributed by overall expression profile. You can grab a hold of a set of tumors and look at whatever makes them interesting to you. PLSR is one where you can, again, distribute stuff, and what you're distributing here are genes instead of tumors, and so the genes location in 2 dimensions is relative to these axes, which are labeled here. So the genes right here, I can mouse over them. These are genes, when expressed highly, are associated with diagnosis of GBM at an older age and low survival, as opposed to younger age and longer survival and so on. And you can grab hold of that gene set and study that and so on. Okay, and I don't have time to talk about the rest of it, but there's a whole lot of other stuff in this set, and it's being expanded more and more. And again, it's open source, and so we'd love you guys to use it. In fact, we'd love you guys to build tabs that could be added to this and so on and so forth. The final thing I want to say is you go back to this thing, and all these lines that look so complicated, and that's what GBM looks like essentially. The vast majority of these things have never been published, right? It's a minority that have publications associating that gene with glioma, and it's out there. It's just like, there it is, and it's recurrent, and what subset of these tumors that these mutations are found in. If you want to study them, it's like free. Okay, 10 o'clock. My time's up.